Bismillahirrahmanirrahim. Today, inshallah, we shall continue our discussion of the kingdom fungi. These are the 5 phylum that we shall briefly mention today and talk about them individually. The first is the Basidiomycota. If you recall, I suggested before that this was a new classification, and these were formerly thought to be protist, but they have greater similarity apparently to the fungi kingdom than the protist, so they are now classified as fungi. Most of these live apparently in water and soil and function as decomposers and parasites. And these are the earliest known fungal fossils. And curiously enough, apparently their spores are motile, as with flagella, as you see before you over here. And they're apparently also unique among fungi in their spores. Now, that's them. That's really all about the Basidiomycota that I'd like to discuss today. The second topic is, the second group is the Zygomycota, the zygomycetes. This you've seen for sure, and these include the common bread mold, whose official name is Rhizopus stolonifer. So this phylum of the kingdom fungi, the Zygomycota, we're going to spend a little bit more time with, because you'll see if you understand these, then you understand the remainder of the fungi phylum. So these are the Zygomycotes, and an example, as I suggested, is the common bread mold, the Rhizopus stolonifer, the genus and species. This is a review. As you can see here, the scientific name is in italics, with the genus being capitalized and the species is not capitalized. And if you were to write this in freehand, you would underline both genus and species. And interestingly enough, and this is, you will see, is also common among many other fungi groups, the bread mold fungus, they produce 2 types of spores. That is, they produce spores when they have lots of food around, and then they produce a different type of spores when the conditions are not optimal for survival. So they have one type of spores that is a result of asexual reproduction, and there's a second type of spores that they produce that is as a result of sexual reproduction. So these fungi produce 2 types of spores. So one of the spores results from a sexual reproductive process, and the second type of spores result from sexual reproductive processes. Now, we're going to study this in some detail. So there are 2 spore-producing sporangiums here, so to speak. This is the first type, and they look morphologically different. That means they look physically different. So this is one spore-producing factory. And then there's a different type of spore-producing factory, which is over here. Now this one is the result over here of asexual reproduction here, and then the sexual reproduction over there. So this is what we're going to study in a little bit more detail, as I said, because the pattern recurs. So what happens? Well, when a spore lands on a bread. Now bread is like, carbohydrate. It's like a party. It's just like the ideal medium for these fungi to grow. So when a spore lands, let's say a spore lands over here, right, on the bread, the spore gives rise to hyphae, and they're called stolons. So the spore grows into hyphae, which are called stolons. That's this over here. And some of these hyphae, they're called rhizoids, or you can remember that rhizoids as roots. They anchor and they go kind of dig deep. So some of these hyphae dig deep, and some of them grow laterally like this on the surface of the bread. So the ones that dig deep into their medium, the bread in this case, they're called rhizoids, and they anchor the plant, and they secrete the digestive enzymes, as we talked about before, and they get the nutrition from the bread. Now, some other of these hyphae, they grow upward like this. They go upward, and they develop into sporangia. They produce sporangia, develop into sporangia, and from asexual reproduction. So they grow into structures like these, and they produce spores over here by a process of asexual reproduction. And you can imagine that's kind of be like mitosis, right? So that's how the basic bread mold pattern is. So when a spore lands on bread, some of the hyphae grow this way, they're called stellons, and some of them go deep like this, they're called rhizoids, and some of them hyphae go up like this, and they produce spore producing factories like this, they're called sporangia, and they produce new spores for the next generation. Now these spores that are produced, those are as a result of asexual reproduction. So here it is, the spore producing factory over here. This is from a common bread mold over here. This is some bread or a cake, whatever it is, and here's the spore producing sporangia. Now asexual reproduction means that the spores are produced by basically a process like mitosis, and these spores develop in the sporangia. And then they are released. So here's the sporangia, and then they're released to the neighboring area, and then some of these would land over here, and they start another colony of fungi over there. Okay, they produce hundreds of these spores. Okay, if they land on moist surface, then they develop into hyphae. Now, as I said, if they land on granite and there's no food around, they're not going to really develop. But as you can see, how a bread mold in one area of the bread can pretty much spread into the rest of the refrigerator, right, in a nearby area at least, because of these spores being so tiny, they just kind of like fly around. And if you open the fridge and this happens to be open, like poof, they go everywhere, right? So these are a result of asexual reproduction. So again, here the sporangium producing these spores, and this particular one landed over here and producing into another hyphae, and then that's the germinating spore. So now sexual reproduction occurs in unfavorable circumstances. That is, if the bread is dries out, or if there's no more food left or whatever, if conditions are unfavorable, then different process occurs and different types of spores are produced, and these spores that are produced are from asexual reproduction. So unlike these tiny spores, the ones that result in difficult situations they're going to be naturally more sturdy, because the idea is to protect them until a time suitable for these to germinate in the future. Maybe the next bread is that shopping spree, and then people bring bread and put it in the fridge, and until that time they produce these sturdy spores. These sturdy spores are thicker walled, and these are called zygospores. Okay, so and the factory produced is the zygospore factory, so to speak, right? So what happens is very fascinating. See, there are different fungi in the same species that, just like there are different people. Like, it's like their human beings are Homo sapiens, but that doesn't mean that every Homo sapien looks the same, right? Same thing with this species, Rhizopus stellonifer. There's one species, but there are different types of Rhizopus. And you say, well, all bread mold look the same to me, but that's like saying all Chinese look the same to you, all Indians look the same to you, or something like that. But they're not. They're genetically different, okay? So, for example, then, if you have like this particular fungal colony and this particular fungus over here, these are two different fungi, and they are genetically not identical. But if they are compatible, okay, then they can have then they can reproduce sexually. So what happens is this. So these hyphae, I'm going to erase some of this so we have some more room. These hyphae that are produced by these tiny spores, they are haploid. All right. So that's the first thing we have to appreciate. Now these spores, they are haploid, okay? And so this haploid spores, right, lands on the bread and produce haploid cells over here. So, and the same over here. Now this fungi that was produced, okay, that too is haploid. So the spores that one produced is also haploid, but they're not identical genetically. So let's put them like a blue N over here. So this one has, haploid nuclei that are genetically different from the haploid nuclei over there. Now, there's no male or female designation over here. So in fungi, most of the time you don't call male or female fungi. So so you call one of these a plus mycelium and the other one a negative mycelium. You could, somebody could say male or female, but it's just different. The concept of X and Y chromosome, it doesn't apply here. So so they're different. So one is designated plus, the other one is designated minus. So the haploid hyphae form from compatible mycelia, okay, as long as they're the same, species, basically. Then the strain fuse. So this hyphae and this hyphae, they come together and they fuse over here. So the negative mycelium, positive mycelium, they fuse together, all right? And they come together and they produce this particular structure, and that's called gametangias and gametes. So gametangia, or coming together of the gametes. And then they fuse together to form a zygote. Now what's happening here is over here the two cells fuse. Here's one cell and the other cell, they come together and they fuse, and there are two nuclei in there, right? There is the red nuclei, and then there is the blue nuclei from the other one, okay? So what happens then is that this one particular cell and fuses and makes one cell with one nuclei, and I'm just like this. All right, ta-da. So the nuclei actually fuse. So you go from a haploid nuclei to a diploid nuclei. You follow? So two haploid nuclei fuse to give a zygote. That's very different. See, two cells can come together and become one cell without the nuclei fusing. We have seen that before, okay? But here the two nuclei fuse, and as a process, as you can imagine, new genetic material is formed. Now, if this particular zygote, if this particular cell were to undergo meiosis, then it would produce four unique haploid, four unique cells that are different from either parent over here, right? So now that's what happens. So this zygote then produces in the new generation that's different from this parent and different from that parent over here. So that's what we mean by sexual reproduction. The definition being that in sexual reproduction, two different cellular material, genetic material come together to give in the next generation, which is different from either parent. So this zygote is formed from hyphae of two different fungi with two different genetic material, and a zygote is formed and the nuclei fuse. And when this zygote germinates, it doesn't have to germinate right away. It can germinate whenever it wants to germinate, when the conditions are optimum. And those cells that are produced by meiosis, that's meiosis, right? Those will be haploid spores again. Okay, now the same process is depicted over here. All right, this is nothing different. I'm just emphasizing this because once you understand this process, we'll see the same process recurs with the remainder of the fungi phylum. So here's the stellans, right? One is colored blue over here, the one is colored green over here. And these are the individual sporangia which ordinarily would give the haploid spores that give rise to all of this. So, but the stellans come together here, the blue nuclei and the green nuclei come together, and they're called positive and negative mating types. And they form this little one cell that's called gametangia, and the cell fuses to produce a 2N zygote or zygospore. Zygote or spore, right? Zygospore. So the zygote is called a zygospore, and the zygospore by process of meiosis gives rise to, here it is, by a process of meiosis, gives rise to a new sporangium and spores, which are haploid again, but these are not the same haploid as the parent haploid spores. So it's a new generation with entirely distinct parents. Now, if you see when these land over here, the spores form, and it's neither green, neither blue, it's kind of like purplish. So I like the color discrimination in this particular picture to illustrate the process. So that was the Zygotomycota, and that's the Rhizopus stolonifer, the common mold fungi phylum. The third phylum is the Ascomycotes. The Ascomycotes, or the sac fungi, which also includes, curiously, the yeast, the common yeast that everybody knows about. Now, sac fungi are so called because they produce their spores in these particular structures, which are called sacs. They're called ascospore, and ascus is basically a sac spore. The morel, which is over here, which apparently is a very expensive fungus delicacy, is an example of an ascospore. So the Ascomycotes are the sac fungi, and apparently this is like the largest phylum with some 30,000 species. And there are two types of spores that they produce. Just like the Rhizopus stolonifer, they produce two types of spores. They produce spores just like before, asexual reproduction, and by mitosis type process. And these are all haploid spores, just like before. And the structure is this. Then their spore producing factory looks different. It actually looks beautifully different in a way, and it's called a conidiophore. So this conidiophore is very equivalent to what we saw before over here, like this. That would be the conidiophore equivalent. The sporangium of the stolonifer is the conidiophore equivalent, right? That's this. But then when the going gets tough, it does the same thing. It produces spores by sexual reproduction, and this occurs in the sacs, which are called ascospores. The sacs that produce these spores are called ascus or sacs. Okay? So now this is the asexual reproduction. Most Ascomycotes reproduce asexually by producing conidia in conidiophores. So this is the whole thing is called a conidiophore, and each little spore is called a conidia. These are just different terms for something, a process that's very, very similar between all of them. So here's a hyphae, and then haploid nucleus, and they divide, and then some of these hyphae form these specialized structures called conidiophores, and they give rise to these hundreds of these spores. That's over here again, conidiophore. Okay, and then there are many different. Obviously, this is a 30,000 rich species rich phylum. So there are many different physical variations to these fungi, but the physiological pattern is very similar. So here's sac fungi. So if you were to take a microscopic view of this particular fungi over here, and you see this particular sac. So this is called a sac. That's the sac we are referring to. It's like a sac that has like a few pebbles in it, like that type of thing. Okay, and compared to the other conidiophore, this one only produces like 8. 1, 2, 3, 4, 5, 6, 7, 8. So 8 spores in each sac. So ascospores are produced in these particular sacs. Now, and as I said, this is a result of sexual reproduction. And when the sacs mature, each of these spores is released individually like this, and to give the rise to the next generation. Now, you will see the process that results in the production of the same is the same compared to the Rhizopus stolonifer, the bread mold. Again, here we have a positive mating type and a negative mating type. These are spores from different fungi, different mycelium, and they're genetically different, right? And here's a red one, here's a green one, right? And see, and then these hyphae, the mycelium, they're so enmeshed in the soil that they themselves probably can't tell the difference between who is who. So anyways, when they come together, so they come together, all right? And some of these positive and some of these negative come together and make one hyphae. So, and then what happens, then they grow over here. And if you look at each one of these, if you were to look at one of these cells over here, all right, if you magnify this over here, you'll see two nuclei like this. One of them would be the red nuclei, and the other one would be the green nuclei right there. See, the red one and the green, each one would have that. And then you will see, as they grow over here, these two will fuse to give one nuclei, which is demonstrated over here, and that's the zygote. Okay? And so the nuclei fuse again from two different members of the species. And once they fuse, they form a zygote. And the zygote, which is 2n, it divides first, it gives 4 haploid, and then they divide each like this to give a total of 8 haploid spores, like this. And you can see the process is extremely very similar. Here they give rise to 8 haploid spores, and each sac has 8 of them compared to what we saw before. The same process is again demonstrated in this picture, so just so that you get the point. And then here we have, instead of green and red, you have a white nuclei and a black nuclei from 2 different members of the species, like Mr. Smith and Mrs. Brown, whatever, or Mr. Smith and Miss Brown, and they come together over here, and then they fuse together over here. This is very fascinating. They fuse into like big bundle thing over here, and then they pair up. The nuclei, amazingly, they pair up like this, one black, one white, like this, and then they keep dividing by mitosis. This hyphae keeps dividing by mitosis, and it has 2 nuclei in every single cell. Great. And then eventually when you form this mushroom, now remember, this whole mushroom is a result of this particular hyphae that came as a result of coming together of these 2 separate members of these species. So mushroom is the fruiting body. It's like the sexual reproductive organ of a fungi. So in the fruiting body, in the mushroom, the 2 nuclei fuse together to give a diploid nucleus like this. It goes meiosis and then mitosis to give a total of 4 spores in this ascus, in this sac. And then when the sac is all mature, and then the spores are released to give the 8 new members of the next generation. And the cycle continues. So this picture demonstrates both of them. Asexual reproduction occurs in the conidiophores, which are like this. The conidiophores, they only have 1 nuclei per cell over here, as you can see. And sexual reproduction occurs when 2 members of two different members of the species come together to give these special cells with 2 nuclei, which fuse to give the zygote, which then divide to give the 8 haploid spores in the ascus, which are then subsequently released. So that's the sac nuclei. That's the sac fungi. Now the yeast are also Ascomycotes, the common yeast, which is used to make bread. Now the reason they are used to make bread is because yeast, when you put them in the batter and when you're cooking, bread or cake or whatever you're cooking, the bread and the cake has sugar in it, the carbohydrates, and the yeast digest the carbohydrates. Now yeast being simple, the byproduct of their metabolism is carbon dioxide, which is a gas, which is what causes the bread to rise. So it's the yeast in the bread that causes the bread to rise, and in fact it is the carbon dioxide that's produced by the yeast as a result of metabolism, carbohydrate metabolism, that gives rise to the gas and fluffiness of the bread. The same process actually also produces is ethanol in different species, which is why it's used to make beer and whatever alcohol people do. See, all living things do cellular respiration, which is taking glucose and getting energy out of glucose. If you remember, the cellular respiration had three parts. The first part was from taking glucose to pyruvic acid, and the second is taking pyruvic acid to the mitochondria, and then from in the mitochondria, the Krebs cycle and whole shebang. But yeast don't have the Krebs cycle, so what they do is they take the pyruvic acid. Either they produce lactic acid or ethanol. So this is called alcoholic fermentation. Okay, some bacteria and some mammals, like we, produce lactic acid. So when there is not enough sufficient oxygen, when there's insufficient oxygen, because if you remember, breaking down of glucose, this will go to the mitochondria over here like this, right? This process requires oxygen. If there's not enough oxygen, like for example, we have a very sick patient and their lungs are not working, and they don't have enough oxygen. Their body does not have enough oxygen, so their Krebs cycle doesn't work. But you have to do something with this pyruvic acid, because otherwise this NADH will keep building up. So what human bodies do is they convert the pyruvic acid to lactic acid, and this is called lactic acid fermentation, basically to rejuvenate NADPH, so this glycolysis part continues. Now, in the yeast take the pyruvic acid and they produce ethanol out of this, and there's byproduct of that is carbon dioxide. For us, we make lactic acid. There's no oxygen available. For yeast, they take pyruvic acid and they convert it to ethanol, and this process requires NADH, regenerating NAD plus over here, so this process can continue. The idea is to keep the glycolysis moving, otherwise there's no energy that the cell can derive from glucose. So yeast are used because of their ability to ferment pyruvate to produce ethanol. And this same process, similar process occurs in human beings, where we take pyruvic acid and produce lactate instead, because we have to keep the glycolysis going. In human beings, if in patients, for example, in the hospital, that they're very, very sick, and if you check a lactic acid level and it's high, it's not a very good thing. It's suggesting that their lungs, or whatever physiologic phenomenon is going on, the disease process requires a certain amount of oxygen, and that oxygen requirement is not being met, either because the lungs are very sick or because the body's metabolism is increased so much that the lungs cannot keep up with the demand of the oxygen. That's anaerobic respiration. It's a bad sign. The fourth category of the kingdom fungi that we should talk about are the Basidiomycota, or the Basidiomycetes, the inhabitants of these phylum are called the Basidiomycetes. Now these are the common mushrooms. These are the mushrooms that we think about. There are apparently some 25,000 species in this mushroom group, and including the common mushroom, the puffballs, and bird's nest fungi, etc. And Basidio means like a club, like, a club that people hit with and stuff like that. So anyway, they have club-shaped spore-producing hyphae called Basidia. So they're called club fungi, the club-shaped spore-producing hyphae. So their spore-producing factory looks like this, and it has 4 spores at the tip. And this is called the Basidia, Basidia. And the club fungi are also responsible apparently for these fairy rings. If you look for it, you will see this, either a complete ring of mushrooms like this, or semicircular rings or something like that. A curious phenomenon where mushrooms kind of tend to go in arcs like this, or sometimes in whole circles like that, and they're known apparently as fairy rings. And in times before, people used to think whatever they wanted to think, that this was a portal for fairies or ghouls or whatever creatures of their imagination. But this is a curious phenomenon that occurs, and this occurs with the club fungi. Now, why this happens is very interesting. See, there was a fungus that started right over here, and its roots started growing in the mycelium, started going in all directions here laterally, just under the surface of the soil, and it kept growing. and things were very good, right? Things were very good. And then because it outstretched resources or whatever, and its survival was threatened, and it was dying. So what happens then is it starts to die. And it grew all of this because it produced lots of spores and stuff like this when the going was good. But if you remember, when the going gets tough for fungi, they produce these fruiting bodies, which are fungi, which are basically where sexual reproduction occurs in fungi. So as the fungi starts to die, that's when it starts to produce these fruiting body. When this fungus starts to die, it produces these fungus, which is where it's a factory for sexual reproduction in the next generation spores. So they're going to die after this. So and then before they die, before these are born, it starts out like this. So the fungus is growing like this. And you'll see at the rim of it is the healthiest mycelium. That's where, and the grass is green over there because of Mycorrhizae, because the fungi is cooperating with the grass. So the grass is actually benefiting from the increased surface area that the fungi are providing. But when the fungi are dying, they produce this rings, and they produce the fungi in these rings because that's the extent of the roots of the original fungi. Anyway, so here's some examples of Basidiomycorys, the smuts that you find apparently. I've seen this too. You've probably seen them too on corn. These are Basidiomycorys fungi. This is called shelf fungi. And Mycelium is a beautiful looking fungus over here. The same phenomenon occurs over here, not surprisingly. As you can see over here, there's a red spore and there's a blue spore, and they grow and they grow, and they have different haploid nucleus over here, and they come together and they pair up just like this, and then they give rise to mushroom, and then the gills of the mushrooms, and right. But if you look at higher magnification, the gills over here at the very tip of them, the two haploid nuclei, they fuse to give a zygote. Okay, so the nuclei here fuse to give the diploid zygote, and the zygote divides by meiosis to give 4 haploid, right? Now this should be technically of different colors because they're all different, right? So this diagram is not as accurate. I hope you appreciate that. And the 4 haploid spores. And this again, the phenomenon is very similar. And again, so the stalk is called a stipe, and then has all these gills. And if you take a cross section of the gills, you'll see a whole bunch of hyphae. So a fungus is nothing other than a mycelium arranged in a particular shape. And at the tips of these hyphae are the basidium, which produce these 4 haploid spores like that. Again, here they had 2 nuclei. The nuclei fuse and the nuclei divide, and each each fused nuclei produces 4 haploid spores. So that's the Basidiomycota. The last of this is the Deuteromycotes or Deuteromycota, and this includes the Penicillium. And there also apparently there's 25,000 some species, and they're unique here because they do not have a sexual reproduction phenomenon. They only reproduce by asexual reproduction. Okay, and this includes the fungi that gives the blue color to the blue cheese. The blue blotches are actually fungal spores that you see. You say, ooh, but then fungi are like plants, and you eat plants, so it's not like, ooh, if they taste good, if they're safe, then you can eat it, presumably, right? So the blue blotches and the blue cheese are actually fungal spores, haploid. Penicillin fungi, which is over here, is also the same thing. It has spores like that. Now, this particular phylum, the Deuteromycotes, are very much have mutualistic relationship with other living things. The lichens that we talked about previously, lichens are fungi and algae, and the fungi in the lichen typically tend to be Deuteromycotes. They are also responsible for a good number of Mycorrhizae, the fungi that participate in cooperative endeavors with the plant roots. So due to mycorrhizae are very much indeed responsible for these two phenomena. Here's again mycorrhizae fungi, increasing the surface area for plant roots for water and nutrients, and benefiting from the sugars that's produced by the plant. Lichens, we talked about lichens, and I suggested two lichens like this, but lichens are, again, a symbiotic relationship between fungi and photosynthetic algae or cyanobacteria, which are bacteria, they're photosynthetic bacteria. So the lichen that you find like crusty growths on rocks and things is one example, but they don't always have to look like this. And there are apparently a great variety of lichens, including this one, which is called the British soldier, because the red coats. The British soldier lichen, the stalks are about they can get 3 centimeters high. And these are still combo living creatures, fungus and photosynthetic algae. And finally, this lichen actually resembles leaves, and you'd mistake it for some kind of a plant. And that's it actually for our survey of the fungi kingdom, and we talked about these 5 phylums. Until next time, as-salatu was-salamu ala rasulillah walhamdulillahi rabbil alameen. Assalamu alaikum.